there's a brief sort of registration. So we will give a little bit of time so people can get their registration in so they can get into the Zoom, and then we can get started.
Take another drink here. There's no beer in it. I just use it for water. That's my true heritage, is Bavaria.
Should we start, Stacy, or we're gonna wait for a minute or one more minute?
I'd say one or two more minutes, because it looks, I can just sort of see the numbers ticking up in the participants. I think people are just logging in.
Okay.
Although now Clint's here, so maybe we can start. But I think just one or two more minutes.
Sounds good.
It's funny, we have done these Zoom calls for a year now, and I still don't know how to turn a pointer on. So I will just use my mouse pointer. Probably better.
Yeah, that should work.
Okay. Well, I think we should go ahead and start. I want to thank everyone for joining today's ARC. We do just remind you to be sure to mute your microphones during the session. At the end, we will have a question and answer, and you can either unmute at that time or put your questions into the chat box. And with that, I will let Renhao Lee do our introduction of today's speaker.
Thank you, Stacy. Let me make sure. Yeah. Well, it's my pleasure to have Dr. Wolfgang Bermeier here as today's seminar speaker. Okay, I've known Wolfgang probably 20 years now. Yeah. Well, I guess we'll start with the real message here. So Wolfgang graduated from college and obtained his PhD degree in Germany, having worked in Bernard Nieswand's lab. That's a premium platelet lab in Germany. After that, he moved to Denisa Wagner's lab at Harvard Medical School, continued his postdoc over there, and continued to work on GP1B and platelets, and then he started with the Galtac thing. I suppose you'll hear quite a bit about in his talk today. After that, he moved to Jefferson University, Thomas Jefferson in Philadelphia in 2007, and then was recruited to UNC Chapel Hill in 2011, and he has since stayed there. And he was promoted through the ranks and become a full professor in 2019, according to his CV. And he, along the way, he has published quite a bit and advanced the field in terms of platelet signaling, especially about the platelet activation mechanism, what happens inside the platelets and leading all the way up to the activation of integrin alpha-2b beta-3. So he has made a tremendous contribution in that regard, plus a lot of other things that he has done. And along the way, he has collected quite a few awards in the field, including the Ash Scott Award, including the Brinkhouse Prize, and he also got the R35 support recently. So without further ado, Wolfgang, we'll be interested to hear what you want to tell us about the signaling.
Well, I would definitely talk about the CalDAC thing, as you call it so nicely. Thank you, Renhao. Thank you for the introduction and the invitation. Obviously, virtual is not the same thing, but it's still fun to meet everybody and to be able to show what we have done over the years and what we are working on right now. Now, I put up a pretty big title as small G protein signaling in megakaryocytes and platelets. Obviously, that's way bigger than anybody could ever talk about in an hour. But it kind of gives me a lot of flexibility, and it kind of introduces what we do in the lab. So we work on all things related to platelets, and that includes the formation of platelets, the activation of platelets, the survival of platelets in circulation, and also obviously their function in pathophysiological situations. And really our main focus is in their classical roles, which is classical hemostasis and thrombosis. So that's what we are right now. That's what we are working on. We do most of our work in mouse models. So most of what you will see will be mouse model work, but we are doing some really, I think, cool translational work, including human platelets. And but most of what I will show you, like really all of what I will show you, is mouse model work. For the talk, I will not talk about megakaryocytes. I will focus on what we mainly did over the last 10 or so years, which is what Renhao already introduced, talking about what makes platelets stick at the site of injury and what makes them actually not stick when they are in circulation, because that's equally important. If they are too sticky, too adhesive, you get thrombocytopenia, and that's obviously a problem. And then also we will put this in the context of what's happening at a site of injury, and this is really classical hemostasis, so mechanical injury, that I will be talking about. Now, I know that most of you are not really platelet people. Renhao, Wilbur, I don't know how many else are there who care a lot about platelets, so I will have an important task here to kind of try and make this digestible for you, and I hope I will succeed in that. We'll see. So anyway, the layout for my talk is to tell you about how important G proteins are in platelet function. And these are the first two sections of my talk. And then at the end, I will spend like 15, 20 minutes to talk about a new in vivo model that we use to study platelet coagulation interplay. So there we get closer to what most of you really care about. And I think there's some really nice eye candy and some limited biology at this point. So this is all in development, unpublished, but I think it will be a really cool model to dive deep into these interactions between the different cell types and the coagulation system at sites of injury. Now, I don't really have to talk much about this. Obviously, hemostasis, hemostatic plug formation. So you have a lesion in your blood vessels. And in fact, what you see here, this little hole, is very similar to what we do in our model systems. You get a hemostatic plug forming platelets as their cellular component, going there, aggregating, forming a thrombus or a plug. And then, of course, you have coagulation happening at the same time. So you have fibrin formation, and all of that leads to a solid, stable plug that is important for preventing blood loss into the tissue. And of course, from the platelet point of view, any sort of acquired or inherited platelet function disorder, which includes low platelet count or thrombocytopenias, can lead to bleeding in these situations. And that's what we are really interested in, both in thrombocytopenias, but also platelet function disorders. And then the flip side, of course, is thrombosis, and there are many forms of thrombosis. Again, platelets mostly being critical for arterial thrombosis, heart attack, stroke. You know all about this literature, but there's clearly a contribution to venous thrombosis and any kind of thromboinflammatory disease like COVID-19, which is obviously on our minds and right now. But I will not be talking about that. Now, to understand platelets, we have to kind of go back and just think about where they live and how they, what environment they are exposed to. Unlike pretty much all the cell types that we normally care about, these cells are not stationary. They are floating around in the bloodstream. They're exposed to really high shear stress situations. And they have to remain in a quiescent, non-adhesive state while they are doing this patrolling of the vasculature. And then as soon as there's a lesion, they have to be able to make contact. They have to make decisions on whether to really make firm contact or not, to build up a plug, how large to build a plug. All of this has to be happening within fractions of a second. And that's really the uniqueness about these cells. And that is also why I think they need a kind of a different signaling machinery compared to other cell types in order to facilitate all of that. And we know a lot about how they do this. It's not moving forward. See, so there are different stages to the platelet adhesion process. There's a stage where the cells are still in the quiescent state. This is called initial adhesion and tethering, and the main receptor for that is a receptor called GP1B59, and I'm sure all of you have heard a ton about that. Mr. GP1B59 is my host today. And so this receptor is important to recruit cells to the site of injury, makes contact with extracellular matrix components, and is really important to slow down the cells and also maybe engage the first wave of signaling. But really the slowing down is probably the more important point here, and that allows typical agonist receptors to engage. These can be of different families. I'll talk about this in a second. And these agonist receptors now induce signaling in the cell. Calcium is one of these important signaling molecules. We'll come back throughout the talk. And there are many changes that happen in the platelet, including morphological changes, secretion of mediators, all kinds of stuff. But arguably, most importantly, fibrinogen or integrin receptor activation. So these are adhesion receptors that are important for the cells to make contact with the extracellular matrix. So bring a first layer of platelets down on the surface, but also let more platelets get incorporated into the plug and therefore form a hemostatic plug or a thrombotic plug if you have a thrombotic complication. For this incorporation of additional platelets, an important feedback system is the release of so-called second wave mediators, and these are lipids like thromboxane or small molecules like ADP. They get released from platelets and activate back onto the same platelets and also newly incoming platelets. And for most of these agonists, what you see here is these wriggly things here. These are 7 transmembrane receptors or G protein coupled receptors. And these are really the key guys in this process. And you can see this also in the drugs that we use in the clinic to prevent thrombotic complications. We have inhibitors to the integrin, the main integrin, very powerful, too powerful, not really used a lot anymore because of the bleeding complications. So now we mostly use inhibitors of this G protein coupled receptor response. And these are drugs like aspirin, which prevents the formation of thromboxane, or drugs that block this ADP receptor, P2Y12. We also have a drug that blocks one of the thrombin receptors, or Apaxa, but it's not really used in the clinic at this point. But overall, this is what has to happen for platelets to make contact. Just to introduce you to how we study this in mouse models, we have a system developed where we use a laser ablation system to induce a lesion in the vascular wall, and this is done in the leg of the mouse. And what you see here in this video on the left is looking down on this blood vessel, and this signal in the middle is a platelet plug that is forming in the blood vessel. And here on the right, we have a 3D rendering of what this looks like. You see the platelets in green, in this case, sitting in the endothelium and preventing blood loss out of the tissue. So we can do this in live mice. We can get quantifiable data for platelet accumulation, fiber inflammation, all kinds of other things we want to measure. Okay, signal transduction. That's when most people normally leave the talk, because signaling is a very complicated thing, and there's a lot of confusing literature out there. In fact, you can find pretty much anything for any literature that you're looking for. Platelets have been studied extensively for signal transduction. However, there are ways to really break this down to a simpler diagram. What you need is an agonist receptor inducing a signal, second messengers to amplify the signal integrators to pick up these second messengers and basically communicate a cellular response. And platelets do this like any other cell type. They're dependent on a certain set of agonist receptors. We have these so-called ITEM or immune type receptors. We have the GPCRs that I already mentioned. In terms of second messengers, the typical candidates, calcium, diacylglycerol, CAMP, phosphoinositides. So these are second messengers used by any cell type that you study. Same for signal integrators, kinases, and small GTPases, and then the cellular responses. I already mentioned integrin activation, but there are other ones that are also important for the function of the cells. Now, as I said, or as Renhao already introduced, we made it a living basically to look into these signal transduction pathways in more detail. We haven't really done too much on the agonist receptors. We did some work, but this is all going back many years. And it's pretty clear that for their classical role, which means hemostatic plug formation, platelets largely depend on the G protein coupled receptors for doing this. And there are several good reasons for that. if you look at the lesion that I showed you before, like a hole in the vascular wall, there are only a few cells that really make direct contact with the vascular wall. And these cells are the ones, or the receptors that mediate the contact with the vascular walls are the immune type receptors. The other cells in the middle, they don't really see the extracellular matrix or any insoluble components. They respond to soluble agonists, and soluble agonists trigger G protein coupled receptor signaling. So to form this three-dimensional plug, you need these soluble agonists, and you need GPCRs to mediate the signal. Another reason is that GPCRs are really fast. So they are basically like switches, meaning that they are turned on and off by a simple nucleotide exchange. And that is really quick, very fast, and is directly connected to second messenger generation. So all of this happens in a fraction of a second, which of course, as I said, is important for platelets, which have to make this decision in a fraction of a second. If you compare this to the immune type receptors, here we have 20 or so proteins that have to assemble into what's called a signalosome. And these proteins have to be phosphorylated, dephosphorylated, assembled into signaling complexes. All of that takes time. And in many ways, this is too much time for platelets really to make the adhesion decision fast enough to form a hemostatic plug. So response to soluble agonists, high-speed signal transduction. And then as a last adaptation that platelets did is, there is also what's called a 1-2 punch system on board, meaning that there is one receptor for each agonist that initiates a signal and another receptor that sustains the signal. So that allows the cell to make first contact, get activated, and decide if there's enough of a need for the cell to stick around or to go on. And only if both signals are triggered at the same time does the cell stay and you form a hemostatic plug. And so these initiating receptors normally respond to smaller amounts of agonists. They're really fast, but they trigger a very reversible response, while the sustaining receptors are a much more solid response, but it takes longer for this to happen. So it's a perfect 1-2 punch system. You take one or the other away, you have a defect in platelet adhesion and hemostatic plug formation. On top of it is negative feedback regulation that gets everything back to baseline and also makes sure that there's no unwanted activation. So that's all on the GPCR level. What our work really contributed is the knowledge that something very, very similar happens on the signal integrator stage. And there we have a different G protein called a small GTPase. In this case, it's a small GTPase RAP1, which is regulated in a very similar way. And I will tell you about this, so I don't go through this in detail, but you have an initiator, sustainer, and you get the same 1-2 punch system. All right, so what are these RAP GTPases? I mean, as I said, they are switches, just like the heterotrimeric G proteins. Activation is mediated by the loading with GTP. Inactivation means that the GTP is degraded to GDP, and the proteins that assist in the loading are called GEFs, or guanine nucleotide exchange factors, while the proteins that help with the degradation of the GTP are GAPs, or GTPase activating protein. This is happening in many cell types, including platelets. In platelets, we know that these proteins are important for integrin function, so adhesion, but also contribute to other things. And this comes back at the very end of my talk. Now, why do I think that this is important in platelets? Well, the first piece of, I think, pretty good evidence is we just have to look at how much of this protein is expressed in platelets. So this is proteomics data from mouse and human platelets. And what you see here is that these RAP1 proteins, there are two isoforms, RAP1A and RAP1B, are expressed at about 200,000 to 300,000 copies. That's about, it's in the top 10% or top 5% of all proteins in platelets is RAP1 protein. So there's a lot of it, a ton of it. The other members or the other family members of the RAS superfamily are expressed at much lower levels, including all the RAS proteins, which are of course very well known for function in cancer biology and other cell types. And in terms of potential activators or GAPs, there's also seems to be a very reduced system on board, meaning that there's one particular GAP and one particular GAP that is expressed at higher levels, and high levels are probably required to mediate the activation and inactivation of a lot of protein like what we have for RAP1. The GAP is CalGAF1, a protein called CalGAF1. The GAP is a protein called RASA3, and I'll talk about those in a second. So what do we know about RAP1? There's a ton of literature out there. A lot of it goes back more than 20 years. There's a very good temporal correlation between RAP1 activation and integrin activation. So these two things go hand in hand. Gil White's lab many years ago made a knockout mouse for the main isoform, the RAP1B isoform. and found that there's partially impaired integrin activation and a defect in hemostasis. So they clearly showed that there's an important contribution of RAP1B in platelet function. All of that led to a model where people said, okay, there's probably a RAP1 dependent and a RAP1 independent pathway that leads to integrin activation that is mediated to a protein called tailin. Brian Petrich was in your division. He was one of the experts in tailin biology. And there's somehow, the idea was that there's a RAP dependent and independent way of activating tailin. Now, I always had my doubts about that, and we finally showed that, in fact, it's pretty much all RAP1 dependent. So we knocked out both isoforms, the A and the B isoform. What you can see is that each isoform is important for activating integrins in platelets. This is an in vitro flow cytometry assay, integrin activation assay. And if you take both isoforms out of the way, integrin activation is down to the level where the tailin knockout platelets would be, very close to it. So no tailin, no integrin activation, or very little integrin activation, no RAP1, same kind of phenotype. We can go in vivo and test this in our mouse model, what I showed you before. And what you find is that in a genotype dependent fashion, you get more and more bleeding at the site of injury. So now you see blood actually coming out of that lesion. So this is where we induce the hole, and there's blood coming out to the left here. There are some platelets that get recruited there. That could be passive recruitment or active. We don't know this at this point, but there's basically no hemostasis going on in the RAP1AB double knockouts or the tailin knockout in. So a very, very important signaling system in order to get platelets engaged and plugs to form. Now, what about CalDAC-F1? This is actually how I got into the whole platelet signaling business. This is now going many years back. And so this is the main GEF expressed in platelets, the main potential activator. When we look in vitro and look at the platelet's ability to form aggregates in a sample tube, we can see that to some agonists, these CalDAC-F1 deficient platelets can aggregate. It's always delayed. So there's always a delay in aggregation that correlates with a delay in the RAP1 activation response. And even if we do get aggregation, there's normally a right shift in the dose response. So this protein is really important to get things going and to provide a high sensitivity to agonist stimulation to platelets. When we go to in vivo, we see a phenotype that is very similar to the RAP1AB double knockout mice I just showed you. But there's more platelet accumulation in this case. So over time, we see some platelets accumulating, and some of these injuries actually occlude with time. Now, keep in mind, these are 50 microns in diameter, very small injuries. If you would do a tail bleeding time assay, these mice would not be able to prevent bleeding from a much larger lesion as seen in the tail bleeding time assay. So CalDAC-F1, really important to kick off the whole system. What about turning it off? So what about this main gap protein expressed in platelets, protein called RASA3? We made knockout mice for RASA3. We knocked it out germline, so in all tissue cells, or we knocked it out in platelets and megakaryocytes specifically. And to our surprise, what we found is that these mice are perinatal lethal. So you get some embryos that are born, but overall lethality at weaning, none of the mice survive. And that is true for both the germline and the conditional knockout mice. When we look at the embryos themselves, what we find is a bleeding pattern in the embryos. And if you look carefully, what you actually see here is a fusion of what you see are lymphatic vessels that are filled with blood. So there's a fusion of the blood and the lymphatic system, or a lack of separation between the two systems. And that's known to be a function of platelets during embryonic development. And in this case, the reason for this mixing of blood and lymphatic system is the fact that these mice have virtually no platelets in circulation. So when we look in the blood of the mice, and this are the few surviving mice we had, there's virtually no platelets in circulation. So losing this gap leads to a thrombocytopenia, a massive thrombocytopenia in the animals. Now, we got lucky to find a collaborator who had a hypomorphic strain. There's a mutation here at the C-terminus of this protein. And in these animals, there is, again, a very dramatic reduction in platelet count. We talk about 95% reduction compared to controls. And the platelets that are circulating are pre-activated. So that we see some sign of RAP1 activation, some sign of integrin activation. Obviously, these are only the cells we can see. The ones that got cleared or that are removed, we don't know. And the reason for the thrombocytopenia in this case is largely due to a problem in survival of platelets in circulation. I don't have the data here, But if you study the half-life of platelets in the mice, this is markedly reduced in these RASA3 mutant mice. And what is also cool is that if you cross the RASA3 mutant mice with the CalDAC-F1 knockout mice, so you have the lack of the gap and the lack of the GEF protein, now you get your survival of the platelets back, and you get the platelet count back to almost normal in these animals. So there's an antagonistic balance between these two players, mediating RAP1 activation, mediating tailin-mediated integrin activation. And this is just to highlight the phenotype again for the mutant mice. So for the CalDAC-F1 deficiency, we have a normal platelet count, but impaired platelet function. Bleeding times are high and thrombosis is low. So these are protected from thrombotic complications. There's also a mild neutrophil defect because the protein is expressed in neutrophils. For the RASA3 mutant mice, it's the opposite. So here we have a very strong thrombocytopenia. Platelet survival is down. There's this blood lymphatic mixing phenotype, and there's a lymphopenia because it's a lymphocyte protein too. Now, what I don't have time to talk about, what's really exciting for this work is that there are now patients with mutations in CalDAC-F1. It's appeared over the last 5 to 10 years in the literature. The disorder is called inherited platelets disorder 18. The mutations all along the gene leads to, in most cases, to a non-expression of the protein. And the phenotype of these patients is very similar to what I've shown you in the mice. So they have an impaired platelet function, not number, and a bleeding phenotype that is similar to Glanzmann thrombocytopenia action. All right, so this is just in a nutshell summary of 10, 15 years of research on how important RAP1 is in platelets. Yep.
So at the Pediatric Institute board meeting was today, and Lucky had asked me to present about the GRA proposal, and also, Peter and B and Vivian presented about the current sickle cell program. So it all went pretty well. I think I made a faux pas, however, because I mentioned the Teresa White chair, and apparently,
Clint. Hi, Clint.
I thought that was something that was sort of just in the works.
Stacy, can you mute him?
Apparently, that triggered a. All right, here we go again.
Sorry about that.
No problem. That's the fun about Zoom meetings. Anyway, so this is just in a nutshell to show you how important that system is. I want to spend a few minutes just talking about the regulation of the system. And so that is work that was in part done with Ren Hao as a collaborator. We were interested on what triggers the activation of these proteins or the inactivation of these proteins, and how is RAP1 connected to the integrin. Is it directly connected, or is there an intermediary protein? And so for RAP1, this is the structure of the protein. You have a business end here with the CDC25-REM domain, and then a catalytic end, catalytic domain, a regulatory domain with a pair of EF hands and a C1-like domain. And it was actually shown several years ago now that these EF hand domains have very high affinity for calcium. So they can bind calcium at a 80 nanomolar, 100 nanomolar range, which is very similar to the concentration actually in a resting platelet. And the resting concentration of calcium in resting platelets is supposed to be around 20 to 50 nanomolar. So a little bit of calcium in all likelihood triggers these domains and triggers an activation of the protein. And that, of course, would explain why it's so important to kick off a response in the platelets, and why these CalDAC deficient platelets are impaired in their ability to form a plug. Now, to go after this and show that indeed calcium binding is important for the function of the protein, we developed a cell-free activity assay. So these are purified proteins, and we monitor nucleotide exchange on the RAP1. If you just put in RAP1 protein alone, there's almost no nucleotide exchange. We add in CalDAC-F1, we see a lot of nucleotide exchange, or a nice increase in nucleotide exchange. And if you use a mutant that is not able to bind calcium to these EFN domains, this is markedly blunted. So the binding of the calcium is really important to get the protein going. And as I said, in collaboration with Ren Hao, we tried to figure out how this works. We needed some structural information, and we used his HDX system to go after the changes that are occurring because of the active or inactive form. So we looked at the active and inactive form of the protein, And what you see is that there are important changes in the FN domains, but that makes sense because, of course, we have our mutations in that domain. There's a linker region between the FN domains and the catalytic domain, and there's a lot of changes there. And then there's also, of course, a difference in the catalytic domain. And all of that supported a model where this linker region is an autoinhibitory loop that prevents the activation of RAP1 or the binding of RAP1 to the catalytic domain of CalDAC-F1. We proved this in a way where we introduced another mutation where we forced the linker out of its potential binding pocket. This is this B46E mutation here. And as you can see, the calcium binding impaired mutant now is not sensitive anymore. It has the same catalytic activity as the wild type form of the protein. So getting the linker out of the way allows the protein to work. And that is shown here in the schematic. So this linker doesn't allow the RAP1 protein to dock in. If we have calcium binding, there's a conformational change, moves the linker out of the way, RAP1 can bind and nucleotide exchange can happen. So calcium is really important to get this protein going. Then the question, of course, was what does the C1 domain do for the function of the cells? This is some platelet data, actually now 10 years old, where we used a retroviral approach to express a mutant form of CalDAC-F1 in platelets. Lacking the C1 domain, there's a reduction in the function in terms of the ability to stick together, to aggregate. However, if we do the cell-free assay and we have the same mutant where we lack the C1 domain, the catalytic activity is the same as the control protein. So it's not really important for nucleotide exchange on RAP1B, which of course suggests that this probably has to do with localization within the cell, and we could show that too. So this is hex cell data, and if you express intact CalDAC-F1 in hex cells, you get a membrane localization of the protein. The delta C1 mutant does not have that membrane localization. And so we were, of course, interested on what mediates the membrane localization. This is the amino acid sequence of CalDAC-F1, the C1 domain, compared to other members of the CalDAC-F family. And you can see that there are some important differences here in the sequence, especially these arginines and the charges next to the arginines caught our attention. And so we did some molecular dynamics simulations and came up with a model that shows that these arginines mediate the interaction of CalDAC-F1 C1 domain with phosphoinositides in the membrane, allowing it to localize to the membrane. We could show this by liposome co-sedimentation assays where we have phosphoinositide PIP2 in the liposomes, and it brings down CalDAC-F1. And if we make those mutations of those arginines, we lose all that binding. And again, in the hex cell, it's the same thing. We lose the translocation to the membrane. So this C1 domain is important for moving the enzyme to the membrane. Now for RASA3, the gap, this is the domain structure of that protein. It's actually known to be a membrane protein in resting platelets. That has been shown many years ago. And the key to that is this pH-BTK domain here at the C-terminus, which can bind to PIP2 and PIP3. That's really all we know at this point about where it is in the cell and how it's regulated. All we know is that that this phosphoinositide environment is affecting the activity of the protein. And I won't show you the data here, it would take too long, but basically comes back to this model. If there's a change in the phosphoinositide environment, we know that then RASA3 is inhibited, and that leads to prolonged RAP1 signaling in the cell. But there's a lot of work that needs to be done there to better understand how this works. Now, the last thing I want to talk about here is how does RAP1 connect to tailin and the integrin? And that is an interesting story too, because from literature in other cell types, especially blood cells, it was shown that RAP1 cannot really connect directly to tailin. It needs an interacting protein. There are several members of that family. The most well-known is a protein called RIME, or RAP1 interacting molecule. So in white blood cells, RAP1 binds RIME and RIME binds tailin. Together, they form a complex that allows integrin activation to happen. Now, the problem is that in platelets, this again proteomics data, RIME or other of these potential connectors are not expressed on a proteomics level, on a transcriptomics level. They're just not there. While we have 200,000 copies of RAP1 and tailin expressed in these cells. And people actually also looked at the RIAM knockout mice and found that there's no phenotype in terms of platelet function in these mice. So this is not an important mechanism in platelets. And so the idea came up that maybe in the platelets, probably for reasons of efficiency and fast activation of the integrin, that RAP1 may be directly interacting with TALEN. And in collaboration with Mark Ginsberg's lab at UCSD and then a group in Germany who worked on the same topic for several years now, it was shown that there are two interaction surfaces for RAP1 in TALEN. This is the TALEN head domain with its 4 firm domains, and RAP1 can bind with the F0 and in the F1 domain of TALEN. And these arginines that are indicated here are important for that interaction. That all has been shown in vitro assays. And it's not a very high affinity interaction, but seems to be higher affinity in a membrane context. Now, what Mark's lab did is they made the mutant mice. So they made a mutant that is not expressing normal TALEN, but this R35E, R118E mutant of TALEN that doesn't interact with RAP1. And what we did with them is to basically compare the phenotype, the integrin activation phenotype in these platelets compared to RAP1AB double knockout platelets, because you would expect similar phenotypes if this is really how the activation happens. And indeed, that's exactly what we found. There is a dramatic downregulation of integrin activation in both of these mice. And there's a dramatic increase in the bleeding time in both models, more so in the RAP1AB double knockouts than the TALENs. And of course, we are trying to figure out what the difference is there. But overall, that brings me to the conclusion of this bigger part of my story here, which is what happens in platelets when the cells have to get activated, when they have to get adhesive. In the resting platelet, the integrin is hunched over, is in a non-adhesive conformation. CalDAC-F1 and TALEN are actually in an auto-inhibited state in the cytosol. RASA3 is sitting in the membrane, preventing any unwanted RAP1 activation and therefore integrin activation to happen. This is how these cells maintain their quiescent form. Upon activation, we know of many things that do happen. Calcium is liberated from intracellular stores or is fluxing into the cells. This leads to binding to the EF domains of CalDAC-F1, activation of CalDAC-F1. There's activation of PI3 kinase and changes in the phosphinoinositide environment. That's important, we think, for recruitment of CalDAC-F1. RAP1 is also recruited or is already in the membrane. And there's activation of RAP1, and then RAP1 can interact with the F0, F1 domain of TALEN, and that leads to integrin activation. I also put the GPCRs here because overall, this is in all likelihood the fastest and most efficient way to get from a change in the environment that the platelets experience to integrin activation. You have 2 switches on board here. The GPCR is a switch-like protein, and RAP1 is a switch-like protein. You still have your signal amplification step in between. You have negative feedback through this RASA3 protein on board. This is probably the most efficient way and the most regulated or the easiest regulated way to control adhesion or adhesiveness of platelets at the site of injury. Okay, moving on to getting coagulation in place. And that's really exciting for myself and for the lab. We are in just like at Emory, a very coagulation-rich environment here, and everybody's talking about fibrin and thrombin and coagulation proteins, which we have largely ignored for many years. And just to introduce this, hemostatic plaque formation obviously is not just platelet aggregation. I talked about this part for 20 minutes now, or 30 minutes now, but coagulation is equally important. So solidify the clot to feed back into platelet activation. Overall, we call this the first and the second wave of hemostasis. But as I said, these 2 things don't work in isolation. Platelets help coagulation to be more efficient, and coagulation helps platelet activation to be more efficient. And so we got excited about this or interested in that because we, in our preliminary studies, we saw that RAP1 signaling is not just important for adhesion of platelets, it's also important for this, what's called the procoagulant response. So changing platelets so that they can recruit coagulation factors to the site of injury. And for that response, a key process that has to happen is the exposure of phosphatidylserine on the cell surface. And that is mediated through changes in mitochondria. The mitochondria permeability transition pore has to open. And somehow this seems to be regulated by RAP1B. An important protein in that process is a protein called cyclophilin D, and I just mentioned this because I will show you some data in cyclophilin D knockout mice. Now, when the PS is expressed on the surface, coagulation factors can bind. Factor 12 obviously is very important in initiating the intrinsic pathway. And together with the extrinsic tissue factor-driven pathway, this leads to thrombin generation. And as I said before, thrombin feeds back into platelet activation, but is probably better known for conversion of fibrinogen to fibrin. And these fibrin fibers are important to solidify the clot. When we have enough fibrin, or we don't want to have too much fibrin, so the fibrinolysis system kicks in with plasmin being generated and fibrin being degraded to fibrin degradation products. So all of this we know largely from in vitro studies. There's very little information from actually in vivo. I mean, we have in vivo data in knockout mice, but nobody really visualizes this in real time. And this was one of the goals we had in this project. So what I will show you is a model that we think can be used to visualize all this in real time in 3 dimensions in mice. And to validate the model, we used several knockout mice, the RAP1 knockout mice, cyclophilin D knockout mice, factor 12 knockout mice, and plasminogen knockout mice. And of course, the factor 12, you should expect less fiber information. The plasminogen, you would expect more fiber information, and that's exactly what we found. Before I get to the model, just to show you how important cyclophilin D and RAP1 are in PS exposure. So PS exposure is measured by annexin 5 binding. This is again a flow cytometry assay. You see a dramatic reduction in PS exposure in the cyclophilin D and the RAP1 double knockout platelets. Most of this defect in PS exposure is due to a defect in integrin activation, because we also see a pretty strong reduction in these tailin mutant mice that don't connect with RAP1 and the integrin. But there's also a component that is integrin independent, at least based on this data here. So our goal for this study was to address 2 issues here. How can we qualitatively and quantitatively assess thrombin and fiber information in hemostatic plugs in vivo? And how can we study the procoagulant function of platelets that by per se don't form a hemostatic plug? So these RAP1 AB double knockout platelets, the problem is they don't even form a plug in the first place. So how can we study those in an in vivo context for their procoagulant function? So this is, again, the model that I showed before, the saphenous vein, where we used the laser system to induce the injury. But instead of just using wide field to get 2-dimensional images over time, we now included spinning disk confocal imaging to get 3-dimensional stacks over time. And we optimized the system to get this so that it looks like a real-time video. These are the images we get. And just to highlight that too, we have worked with a really great guy at the math department at UNC, David Edelsteinson, who wrote a software package called Image Tank that we used for the analysis of these videos. And the great part about it was we could work with David. Every time we had an issue, he would fix the code and give it to us for free, and the whole software is for free. So if you do high resolution imaging, contact David. He is more than happy to work with you. Anyway, so this is what this looks like. This is a genetic tracer mouse where we have the endothelium labeled in red and the platelets labeled in green. We have 3 different views. One is a top-down view. So we're looking down onto the saphenous vein. We're looking from the side into the saphenous vein. And we're looking from within the saphenous vein up to the luminal surface of the endothelium. And when we now do the ablation, what we see is that we get the 3-dimensional clots forming in the lesion, just like you would expect. Most of the platelets are actually on the extracellular, extraluminal surface of the endothelium. But we get perfect pictures for that process happening. If we have a 2-color image like here, we need about 10 seconds to assemble a stack. So we have some loss of temporal resolution, but overall it's minimal. We get highly quantifiable data out of it. Now, obviously, more interesting is to look at what's happening on the side of fiber information. So now this is a video where we look at platelets in green, phosphatidylserine exposure in red, and fiber information in blue. And what you see here is that initially there's actually great colocalization between fibrin and PS around this hemostatic platelet plug, actually. But then over time, the PS exposure spreads over the endothelium while the fibrin is retained close to the platelet plug. And that is very consistent. Every time we do this, it's very consistent with earlier studies we did. The fibrin really is there to keep the plug in its place. And that's why we have this ring around the platelet plug. And the PS seems to be, initially at least, helping with the fiber information in that area. So there's a limited localization, co-localization between phosphatidylserine and fibrin during the process. Initially a lot, later on, it's not really co-localizing so well anymore. All right, so how faithful is that system in reporting thrombin generation fibrin formation? So the first mice we looked at were these cyclophilin D knockout mice, and I showed you before, they don't do PS exposure properly. And so we did that. We looked at PS exposure, again shown in red here, platelets in green, fibrin in blue. And when we quantify this, we see that basically all of our PS exposure response is gone in the cyclophilin D knockout mice. So there's very little, there's still a little bit, but very little PS exposure that is still happening. Didn't really affect platelet accumulation much. Overall, the amount of platelets seems similar. But when we looked at fibrin formation, we saw a pretty marked reduction in fibrin formation, not so much in the early stages of the process. We think this is largely due to the tissue factor triggered initiation of the system. But then as soon as the intrinsic pathway has to kick in, we see, or we think we see a reduction in fibrin formation because we don't have PS exposure on either the platelets or the endothelium. At this point, we didn't really know yet. I'll answer that in a minute. Also, what we see is there's more rebleeding in these injuries. So these, in the cyclophilin D knockout animals, we see these plugs disappearing and then forming again more frequently than in the controls. But just to clarify this for the analysis, the quantification, the only injuries are included that don't rebleed, because that obviously could change the system. So we only use injury that form a hemostatic plug that is stable from the beginning. Now, factor 12, we would expect something similar, right? So this is the intrinsic pathway. And indeed, we got a pretty significant reduction again in fibrin formation. Again, initially, kinetics look at least very similar or somewhat similar, but then it peters off really quickly. In this case, it looks to be even less than in the cyclophilin D knockout animals, but overall comparable in the sense that we have a dramatic reduction in fibrin generation. Again, platelet adhesion was not affected in this system either. Now, the plasminogen knockout, you would expect the opposite, and it's exactly what we found. So in this case, we form fibrin faster, and there's more fibrin overall. It spreads more over the endothelium, but it also is more three-dimensional. I don't really have the stack here, but it's also more three-dimensional around the hemostatic or the platelet plug. And you can see that there's even a dose response, so the plasminogen heterozygous seem to fall in the middle here. So you need a good amount of plasminogen to prevent fibrin formation from happening. And what's really cool is when you infuse recombinant plasminogen, here labeled in red, into wild-type mice and see where it goes exactly where you would expect it to be, where the fibrin is generated. And in fact, it spreads out over the endothelium, similar to what we find in terms of where the fibrin spreads in the plasminogen knockout mice. So the plasminogen gets recruited there and prevents fibrin formation from spreading across the endothelium. Okay, so means the model seems to be faithful, seems to be good for what we wanted to do with it. So the question is, okay, is it the platelets that really are important for triggering this intrinsic pathway, and how can we study that? And because we only had access to these germline knockout mice, and so we used a trick that we developed a couple of years ago, where we have a transgenic mouse model where we can inject antibodies into these mice, and that leads to pretty much immediate platelet depletion. And basically all the platelets in these mice are depleted within 10, 20 minutes. And then we can transfuse platelets of our liking into these mice and reconstitute, at least to a certain level, in the blood, the platelet count with either wild-type platelets or in this case, cyclophilin D knockout platelets. So what we generate is basically a system where we only have platelets that are deficient in cyclophilin D, or we have a wild-type situation in this case. So we can ask the question, is it platelet cyclophilin D that is important on platelet PS exposure? And it seems to be the case. So when we do the analysis now, we get a very similar result to the data I showed you before. So lacking PS exposure in platelets only leads to reduced fibrin formation due to a defect in the intrinsic pathway. So that answered one issue. The other one is, how can we study now the RAP1 deficient platelets which actually don't form a plug? And that's a little harder to explain here, but that's a study. We used a trick that we developed a couple of years, or 2 years ago now, published 2 years ago, And that was actually a transfusion medicine paper that we did, where we looked at the effectiveness of transfused platelets in situations of platelet function disorders. And in this case, what we showed is that the endogenous platelets, again in the mouse model, the endogenous dysfunctional platelets get recruited by wild-type platelets into sites of injury. So they make it, the wild-type platelets basically allow them to get recruited to the injury, even though themselves they wouldn't do it. And the key to that is Reinhardt's receptor GP1B, which allows these cells to dock with the wild-type platelets, and then they get basically cemented into that hemostatic plug. It does have an effect on the integrity of the plug. So these mixed population studies show that there's often more bleeding in these situations, but the recruitment is still happening. And so we thought if we can do something similar in our model, we could probably get these RAP1 deficient platelets into the clot with either wild-type platelets or cyclophilin D knockout platelets. I know this is a little hard to explain here. Initially, we just tried the system with wild-type platelets and cyclophilin D knockout platelets. You can see this here. They both go in, labeled in red and in green, and we can monitor fibrin formation. What's really cool about it is we can establish a ratio between the cyclophilin D platelets and the wild-type platelets that is required to get all the intrinsic pathway activity back. So if we do a ratio of 3 knockout platelets to 1 wild-type platelet, we get an intermediate fibrin formation response. If we do 2 to 1, we get pretty much all of our activity back, right? So we know that this is a key ratio, 2 to 1, and now we replaced the wild-type platelets with the RAP1 platelets, because the cyclophilin D platelets cannot really support fibrin formation. That allows us to study the procoagulant activity of RAP1 platelets. This is shown here. Again, both of them get incorporated. So RAP1 platelets in the presence of the cyclophilin D platelets get incorporated into the platelet plug, and we can look at fibrin formation. And if we quantify this, we see that there is a reduction in the if we use RAP1 platelets versus wild-type platelets. So there is an important contribution of RAP1 dependent phosphatidylserine exposure in platelets. However, it's not as strong as we expected it to be. We expected it to be similar to cyclophilin D platelets alone, based on our in vitro studies, but that does not seem to be the case. And of course, that's what we are working on right now. So the summary of this part is, I think we established a 4D imaging model that allows us to study platelet coagulation interplay during hemostatic plug formation in vivo. I think this model will be really helpful to open many questions on this topic that came out of in vitro studies, also clarify a lot of things. We provide the first in vivo evidence that RAP1 signaling affects hemostatic plug formation independent of its key role in platelet adhesion. We found this disconnect between the procoagulant activity of RAP1 platelets in vitro and in vivo, and of course we study why. And then of course we have a lot of work going on looking at how RAP1 actually really communicates with this PS response, and I think other small GTPases will be the key to that. And with that, I think at the end of it, there's a ton of people to acknowledge. Just want to acknowledge some former members of the lab, especially Gio Stefanini, Aaron Cook, who did a lot of the work I showed in the first part of my talk. And then right now, David Paul, Bobby Lee, and Abby is a graduate student who established the 4D imaging platform. She has done this within a year, so that's really phenomenal. And then a lot of collaborators at UNC and people outside of UNC, including Reinhardt, of course. And with that, and thank you to the NIH, of course, for the funding. Thank you.
Wow, thank you, Wolfgang. That's a tremendous amount of information.
Sorry, I may have overdone it a little bit.
That's okay. So we're open for questions. You're welcome to raise your hand or unmute yourself or type in your question in the chat box. While they're, I guess, formulating their questions, I have a few. I guess I'll go ahead and ask you. We have not talked about science for a while, I guess. So RASA3 knockout, right? That essentially literally has no platelets. Your GalDAC knockout, platelet count is normal. Since they're in a pair, like a pull and tug pair, right? Focusing on this part. My understanding is that RASA3 is probably a brake, and the GalDAC is kind of accelerator. In a way that strongly implies this dynamic equilibrium between, I guess, within your small GTPases somehow leads to clearance or leads to activation of the platelet.
Right.
Right. That's my understanding. So have you thought about then what would be the link between this activation of the small molecule, small GTPase, to the clear me signal? Have you explored that part?
We did, and it's in all likelihood, actually the simplest of all explanations, which is there is some integrin activation. And so we find these platelets mostly in the spleen. They seem to be in a loosely aggregated form, and that's all we can say. And the reason why I say integrin, two reasons: A, we crossed the mice, the RASA3 mutant mice, with tailin knockout mice. That saves the platelet count or rescues the platelet count. And we injected inhibitory antibodies to the main integrin, and that also brings the platelet count back up to at least a certain level. So it seems to be largely integrin dependent. We looked at glycosylation patterns on the platelets that didn't seem to be much different. We looked at platelet neutrophil, platelet leukocyte aggregate formation that seemed to be much different. PS exposure is not different. So the most likely explanation is integrins.
Okay. So I guess then the follow-up question is what recognize those things in the spleen then? Recognize the activated integrin. But
Yeah.
That's something to think about. Moving on to your 4D model, do you or can you see stained fibrin in the platelet plug?
Yeah, not really. But we have had issues since we started that 4D or 3D or 2D, whatever we did, at least with our detection threshold that we have, we cannot see fibrin in the hemostatic plug itself. It must be very low levels, or it's not there pretty much at all. And
have you tried to stain for fibrinogen then?
So we did infuse labeled fibrinogen, and it's the same pattern. So there's a lot of interesting biology there that we haven't exploited yet, but it goes, it's consistent with the literature in the sense that if you do, like, I mean, I was in Denise's lab, she looked at the fibrinogen knockout mice in a thrombosis model, right, in the ferric chloride mesenteric model. And the clots formed just fine. They actually formed faster than wild-type clots, but they were not anchored to the endothelium. So they would build up and roll off, and build up and roll off. So in an in vivo context, you do not need fibrinogen to get platelet aggregates. I think that's more of an in vitro artifact. And it's likely VWF, other ligands that come into play there.
Wow. Well, then, I guess a related question here would be, have you checked or do you have the resolution to see the PS exposure on the platelets that in different parts of that platelet plug?
So yes, we do have the resolution. And so the platelet plug, right? And that's what people, I think that's in part the problem. Sorry, Wilbur, I think, is on the call. I mean, microfluidics chambers have kind of distorted the view of how hemostasis works, in my opinion, because in a microfluidics chamber, you have a coated surface of collagen and you have buildup of a clot, right? And there's a lot of PS exposure because you have this coated surface of collagen. But in a plug, really what you have is a hole where only these cells around here make contact with the collagen. The ones in the middle, as I said at the beginning, they don't have contact with the subendothelium. But you need that contact for your PS exposure response. And the soluble agonists are not strong enough to trigger PS exposure. So we find the PS exposure at the edges where the platelet plug intersects with the endothelium.
Yeah, I guess it does give, me a lot of things to think about once we see this part.
Right.
Any other questions from the audience? If they can unmute themselves.
Yeah, this is Pete Lawler. I have a question about factor 12 deficiency. It's not associated with the bleeding phenotype in humans. but it is associated possibly with the thrombotic phenotype. So why do you think in your model factor 12 is important for hemostasis?
Yeah, I mean, so these small clots still form. It's not like they don't form. We do see a reduction in fiber inflammation. That is correct. I get this question. It's the second or third time now that I get the question. I don't have an answer. All I can say is that the system seems to faithfully report fiber inflammation, and it's reduced. I really don't have a great answer for you. But that's actually a good thing, right? So that makes us think about what is different between factor 12 and factor 11, or factor 12 and I don't know.
Another question I have along the same lines is in hemophilia, when template bleeding times are done, where they make an incision in the skin of human hemophilia patients, there is histopathologic abnormalities that are obvious in terms of what the fibrin deposition is. And I'm just wondering, in your animal models, have you looked at hemophilia factor 9 or factor 8 deficient mice?
Yep. So we did that in our first description of not the 4D system, but the regular system that I used earlier in my talk. And we used the factor 9 knockout mice. And it was actually interesting. So the small injury model is largely a platelet-based model. So you get your bleeding when the platelets don't work. If coagulation is not optimum, or actually if coagulation doesn't happen, you still get a platelet plug that seems to be big enough to prevent blood loss, right? But what we did see is that there was a reduction in platelet accumulation, meaning that there wasn't enough thrombin generated to recruit all these platelets. And in the factor 9s, actually, there was an increase in fiber inflammation, not a decrease. In the low tissue factor mice, there was a decrease in fiber inflammation. In the factor 9, there was an increase, which I think, from my naive view of the field, probably is because of what you see in the patients, right? It's not really a true bleeding. It's more of a oozing of plasma into the tissues or into the joints. And probably something similar goes on there, that these plugs are just not as tightly packed, and that allows plasma to get out of the blood vessel. And that's where the tissue factor is. So you get more and more tissue factor dependent fibrin generation in the situation where you have factor 9 deficiency. Would be great to get your feedback actually on that, because that's the only way we could explain it to ourselves. Did that make sense?
Yes.
But bottom line is, we looked at it. The model does report a deficiency in plug formation, but it's not leading to bleeding if you have such a small injury. Now, but you can, let's say, you can sensitize the system to coagulation factor deficiencies by partially inhibiting platelet function. If you treat the mice with aspirin or a P2x12 inhibitor and then have a factor 9 deficiency or a tissue factor low situation, they bleed a lot. So both of the systems have to work together.
Cool. Any other questions? That's a good question. I think.
Hello.
Go ahead, Curtis.
Hey, sorry. My computer has been acting up, so I've caught most of the presentation. So I hope you didn't address this topic. If you did, I sincerely apologize. But from what I've heard, it was extremely interesting. And what my question really pertains to other models of bleeding like malaria. So do you see any relationships between factor 12, or have you guys studied any like infectious disease model like malaria and see if this factor contributes to disease pathogenesis in any type of ID models?
No, we have not gone there. Sorry for that. No, we haven't. I mean, that's the short answer. We haven't really done any infectious disease models. We'll go and start working on staph infection, and we have some colleagues here interested in that, but we have not looked in malaria.
Really nice talk.
Is there a documented change in platelet reactivity and or coagulation in malaria?
Yes. I think is for, I can't remember which factor it is. So I'm an immunologist and I study leukemia. So I just like to read different things, but I have come across a paper that shows like there are clotting issues associated with making the disease pathogenesis more aggressive in malaria. I just can't remember which factor it is that's been associated with that. But it just, your talk made me think of that. I just wonder if there is any relationships between factor 12 and kind of disease pathogenesis, given that clotting seems to be associated with more like worse prognosis, so to speak. So I was just wondering, have you guys looked at that relationship?
We haven't, and I'm really not a factor 12 specialist, so you got to talk to somebody else.
Thank you.
You're welcome.
I think that's our final question for the session. I wanted to thank you again, Dr. Bergmeyer, for an excellent talk. And of course, thank everyone in attendance for their engagement and their questions. And we will close the session. Thank you all.
Thank you.
Thank you, Wolfgang.